Welcome everybody to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Dana Shaw and I'm a postdoc at Washington University School of Medicine in St. Louis. And I'll be moderating today with Joseph Campanale, a postdoc at the University of California, Santa Barbara. We're excited to highlight the work of our outstanding postdoctoral members. And today we'll be hearing from Dr. Daniel Medina Cano from Memorial Sloan Kettering Cancer Center and Jiae Lee from the University of Washington. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. Please enter your questions in the Zoom Q&A box. And I'm excited to introduce our first speaker, Dr. Daniel Medina Cano. Daniel is especially interested in the genetic and molecular causes underlying neurodevelopmental disorders. He began his work as an intern for the Molecular Biology Institute of Barcelona. He then moved on to his graduate work at the University of Paris Diderot in the labs of Dr. Vincent Cantagrel and Dr. Lawrence Callow, where he studied a neurodevelopmental disorder of unknown pathophysiology caused by mutations in SRD5A3. He is now in his postdoctoral position in the lab of Dr. Thomas Verbuchen, and Daniel's main project, which is funded by a New York STEM Training Award, focuses on using genetically diverse mice to generate neural organoids to better understand the genetics underlying complex neurodevelopmental disorders. With that, I'd like to introduce Daniel's talk entitled Dissecting the Impact of Genetic Variation on Gene Regulation During Early Development Using Mouse Forebrain Organoids. With that, take it away, Daniel.
Awesome. Thank you so much for that nice introduction. So first of all, I want to thank for the introduction once again, and thanks the organizer of SDB for giving me the opportunity to show our work to you, and hopefully you will like it as much as I do. Where basically what we try to understand is how the high diversity in brains that we can observe in mammal has emerged through evolution, or said in another way, how like a relatively common set of genes has been used in different ways, such as to give rise to these really diversified structures in the brain that underline also like more fine-tuned differences, such as how the neurons communicate, how the cells act, and as a whole will lead to the more complex of phenotypes that one can observe, that is behavior itself. So actually one of the best ways that one can control through evolution how the same gene is used in different cell types is through changing the regions of the genome that control its expression. So through variation in cis regulatory elements, we can change when, how much, and for how long will a gene be expressed in a certain cell type. So examples of these cis regulatory elements are enhancers, insulators, and promoters. And as you can imagine, if in a certain cell type we are to have a functional change that will affect how these elements will behave, it will greatly impact the way that a gene is expressed in a certain cell type. And this kind of variation in cis regulatory elements that happens across all of the species will have a particular effect when it comes to development. Whereas, as you can imagine, a small change in how a gene is expressed during development in a certain cell type could potentially lead to a cascade of effects that will lead to the phenotypic diversity that we can observe in the adult individuals across the species. However, little is known about how this variation that we all have in these cis regulatory elements changes the way that genes are expressed and could affect and shape development as a whole. So actually one good way that one can study these questions is through the use of F1 hybrids. So in this paradigm, you mix like two individuals from different species, and what you would get would be cells that contain one chromosome from the species A and one chromosome from species B within the same cell. And this is important because any external factor is buffered by the fact that both chromosomes are exactly within the same cell. So any difference that you will be to observe between those two chromosomes, between how each one of the two alleles for a certain gene are expressed, is likely to be caused by changes in the cis regulatory elements that those chromosomes have that make each species unique. So, for example, if we were to use this F1 hybrid cell in a developmental paradigm, so through the differentiation of a cell type into diverse cell types, what we would expect is that both alleles for the same gene would be mostly used in the same way across cell types, meaning that both alleles are used for a certain gene in a cell type or they are not used at all. But it could also happen that in some instances, we would find some cell types that only express the allele from one of the two species. And this is quite likely to be caused by variation within these cis regulatory elements. And as said, those changes could have a major impact on the behavior of that cell, on its phenotype, and the downstream consequences on development on that change. So this is a great tool to understand how this is happening. The downside, though, is that species by definition cannot be hybridized. We cannot do these F1 hybrids. So what we do in our lab is to take the closest that we can get to a high genetic diversity across like subspecies, while we can still hybridize them. And that is happening across different mouse strains or different mouse backgrounds. So here I just show you the classical black 6 mouse, Castaneus, Molf, PWK, and Spretus. And what is really interesting is that when we are to compare those different strains or even different mouse species, we'll find a high degree of variation. So just as a number, if we are to compare spread versus black 6, they have 60 million polymorphisms that make each one of them unique. Or said in another way, like if we are to compare 2 random human individuals and look at 1,000 base pairs in the genome, we will only find 1 polymorphism. If we are to do the same between spread and black 6 and we look at 1,000 base pairs, we'll find 10 polymorphisms, so 10 times more genetic variation than the one that can be observed between humans. And that can be said in terms of like genetic variation, but also like how apart are they in number of generations. So spread and black 6 are differentiated by 8 million generations, whereas humans are different from gorillas by like only 0.6 generations. So as you can imagine, this huge degree of diversity affects anything or any phenotype that one wants to study, starting by like more broad characteristics. So for example, spread mice compared to house mice or black 6, they're so much less aggressive. Like for example, when males have to divide the territory, they would never fight. They will do some rituals to separate the surfaces. They're really good climbers, but they're really bad jumpers. They tend to live out like 1.5 years on average, whereas like, as you know, mice tend to live black 6 much longer than that. And these are like general characteristics, but of course, this genetic variation will also affect any other aspect, and it will, for instance, change how the brains develop. As you can see here in this image, this is a coronal cut of the hippocampus of a black 6 on top and a spread at the bottom. And you can appreciate that there are major differences on how has this structure developed. But what is even more interesting is that if we are to look at the same cell type in this structure, for example, the hippocampus, we'll find difference in the genes that these strains are expressing. So as you can see here at the right in a spread, this is an in situ hybridization against a serotonin receptor, HTR1A. And hopefully you can appreciate how in a spread, we can see that this population here at the hippocampus expresses this gene. In black 6, there's no expression of this gene. So huge variation at any aspect that you want to study, molecular phenotypic behavior. And we want to use this as a model to understand how this variation affects serious regulatory elements, affects gene expression, and changes the way that those brains are formed. So as said, the great thing about these 2 different strains is that they can actually be interbred. So we can do F1 hybrids from this spread and black 6. The downside, though, is that, of course, it's not easy at all. Setting up those breedings takes months. The black 6 always has to be a female. So it's really complicated in our case where we want to study how this variation affects the development to take an in vivo approach. And that's why instead of going in vivo and waiting for those pups to happen, we go directly in vitro to try to make this kind of study so much more high throughput. And for that, instead of using the embryo as a model, what we use is that its closest counterpart in vitro, and that is the mouse epiblast stem cells, or EPSCs. That is a type of stem cells that we've optimized its culture in the laboratory that can be propagated for many passages, and is actually one of the closest equivalents that we have to human stem cells from the mouse. As you can see here below, they actually keep on expressing across many passages pluripotency factors. And then the idea will be then, once we have these epiblast stem cells, just differentiate them into whatever tissue we want to study the F1 hybrid gene. So altogether, this leads me to the main 2 parts of our project, within which first we aim to generate that zoo of F1 hybrid stem cells and optimize the tools that will allow us to map cis regulatory variation and how does it affect to the way that the alleles are being used in a certain cell type. And then once we have this system up to speed, the idea is to come up with a protocol that will allow us from those mouse epiblast stem cells to generate the closest that we can get in vitro to a brain, that is this 3D brain organoid. So these are the 2 parts, but of course, no one does anything alone. And I'm lucky enough to count with the help of Tarek, Marty, Jiae Lee, and Serena across all of these projects. And I just want to say that it's thanks to them that the project is where it is nowadays. So then the first part, to generate the zoo of F1 hybrid epiblast stem cells. How have we done that? So for that, we cross a female black 6 with a male spread, PWK, MOLF, or GAST, and obtained the F1 offspring. From those embryos, we have extracted the fibroblast, and those fibroblasts have been reprogrammed into stem cells, into induced pluripotent stem cells that we have then differentiated into EPSCs. leading us once again to this paradigm. In each individual cell, we have one black 6 chromosome, one spread chromosome, one allele from each one of the two strains. And what is important is that this environment is completely controlled because both alleles are subjected to the same transacting effects. So any difference will be because a cis-acting type of effect. So what to expect? What do we expect when we use this kind of stem cells in a differentiation approach? So the first thing is that for most of the alleles, what we would expect is, for most of the genes, we would expect that both alleles will be equally expressed, they will be equally active, and will use a similar battery of cis-regulatory elements. But there will be some instances when which out of these 60 million polymorphisms that set them apart will fall within a cis-regulatory element and will change its functioning. When this is to happen, when in the black 6 or in the spread allele, we have a polymorphism that changes how an enhancer, promoter, insulator is being used, that will change the way that the gene is expressed, and therefore will lead to an imbalance in the gene expression. So basically, what we aim to do with this paradigm in kind of a broad two-step procedure is first to identify, well, thanks to the fact that all of these mRNAs are loaded with polymorphism, we have to identify those allelic imbalances by looking at how the genes are expressed in certain cell types, if from both alleles or only from one of the two alleles, and try to correlate that with imbalance in the activation of these cis-regulatory elements. So this is the broad idea, but before getting into too complicated, like how would this work in a brain and so on, we wanted to already test if this cell system was suitable for our purposes at the iPSC level directly, so at the pluripotency stem cell stage. And for that, we perform bulk ATAC-seq in those black 6 spread hybrid epiblast stem cells. And just as a background information, ATAC-seq is basically a technology that looks at how open or accessible are certain regions of the genome, usually cis-regulatory elements. So basically what you can see in this plot here in the center, when it comes to like the dark green dots, those represent regions of the genome that are equally accessible or open from the black 6 and the spread allele. But actually the interesting ones are those light green dots at the left or at the right that actually represent regions of the genome or cis-regulatory elements that are more open on the black 6 allele or in the spread allele. And then when we quantify this behavior, as you can see here, we found that up to more than 20,000 regions of the genome are more accessible from one allele or the other. So a huge amount of diversity of like how do these strains activate cis-regulatory elements during pluripotency. And this was actually true also when we were to look at the transcript level. So as you can see here, same type of diagram, same type of idea. Out of 15,000 RNAs that were detected through bulk RNA-seq on these hybrid iPSCs, we found that almost 5,000 transcripts were more transcribed from one allele or the other. So more from the spread here at the right or more from the black 6 allele here at the left. So altogether, giving us this wide library of like thousands of genes and thousands of cis-regulatory elements that are differentially used from the spread or from the black 6 allele. So then this is great, but what have we done with this information? So there are like thousands of genes, but in our case, something that came like jumped right into us when we saw that list of genes was NANOG. So NANOG is a gene that is really important for the maintenance of the pluripotency in a stem cell level and during the embryo as well. And it was kind of shocking to see that actually the enhancer that controls expression of NANOG in the iPSCs was not equally accessible or equally open between the black 6 and the spread allele. So as you can see here in red, the enhancer that is controlling NANOG expression in the spread is much less open. And this was interesting because this was correlated also with a major reduction in the abundance of the transcript of NANOG coming from the spread allele, suggesting that because in the spread that enhancer is less accessible, that leads to a reduction on NANOG expression. So then the next thing that we did was to actually, we do not only have the black 6 spread hybrid, but we have the CAS, the MOLF, and the PWK. And we performed the same kind of analysis across all of these lines. And what is interesting is that, as hopefully you can appreciate here, only the spread allele showed that major reduction in accessibility of the NANOG enhancer. So this is kind of a good way of like pinpoint what individual variant could be causing that. Because the next thing that we did is to look at the variants that make these enhancers unique. So here I'm just showing like a amplified image of this enhancer. As you can see here across alleles, we can find different polymorphism across the species, across subspecies, sorry. So for example, we can find these ones in purple that are shared between the spread and CAST, or these ones in orange that also share. So these are likely to not to cause the phenotype that we're observing in NANOG expression. But among the three that are only happening within the spread allele, we can find this one here that falls within an OCT4 binding motif that is usually observed in most of the strains. And basically what happens in the spread allele is that that OCT4 motif is not functional anymore. As you can see here, we have a change from a C to an A. That makes that motif not functional anymore, leading us to a hypothesis that because in the nanoc enhancer, the spread do not have this OCT4 binding motif, OCT4 cannot bind to enhancer, cannot make it open, cannot make it accessible, and leads to a reduction on nanoc expression. And this is just preliminary data, but we've been trying to measure enhancer activity of the BLAC6 allele and the other alleles. As you can see here in this bar, when we are to compare the wild-type BLAC6 enhancer versus the exactly same sequence, but only with that polymorphism in the OCT4 binding motif, we can observe that there's a major reduction in the activity of this enhancer. So and indeed that it's quite likely that this SMP is causing a decrease in nanoc expression within a spread. So this is great. Obviously, altogether for the first part, we've already come with this panel of F1 hybrid stem cells that we've already used to map thousands of genes and disease regulatory elements at the pluripotency stage. And we actually can take it to individual polymorphisms that may be changing the way that different subspecies express a certain gene. But as I was telling you at the end, my goal is the brain. Like I want to apply exactly the same cell lines and exactly the same paradigm, but to a brain-like type of tissue. But before getting in any further into how is this built, I just want to tell you or remind you how do we go from an embryo, what the iPSCs are resembling, into a brain. So the epiblast stem cells, their closer counterpart in vivo in the mouse embryo are the embryonic day 6, those stem cells within the embryo. And basically these stem cells, the first decision that they will take is to what layer will they become, whether they will become ectoderm, endoderm, or mesoderm. And it will be within the ectoderm that will observe the apparition of the neural plate. And in the more medial part of the neural plate, we'll have the neuronal ectoderm. That's the one that will give rise to the brain. Once we have this neural ectoderm, there are like two main decisions that have to be taken before we reach that cortex type of tissue or the forebrain. So the first one is whether if that neural tube will become anterior or posterior. It's actually only in the anterior part where we will find the forebrain, so the future cortex. And I mean, there's a lot of studies, many in vivo about what signaling pathways are controlling this process. But there are three molecules that seem to be key, or three pathways, such as Wnt, FGF, and retinoic acid, that seem to be potent posteriorizing factors, meaning that if these pathways are to be highly active, we are more likely to lead to a posterior type of fate. Whereas if they are not active, or they are even actively inhibited in this anterior part, we'll have a much more forebrain-like type of fating. So this is to reach the forebrain, but even once we are in the forebrain, there's yet one more decision that has to be taken as to whether to become dorsal or to become ventral. As you can see here in this image, is the dorsal part of the forebrain that will become the cortex. And this is when the things get complicated, because I was telling you that on the first part to become anterior, we need to inhibit Wnt, basically. But on the second phase, we actually need Wnt within those cells so they can properly acquire that dorsal fate. So it's a complex system, but we use all of this knowledge to come up with a new protocol within which, starting from mouse epiblast stem cells, for the first 2 days, we subject them to a high Wnt inhibition to anteriorize that neural plate into anterior neural ectoderm. And then after those 2 days, we change the condition and we add a lot of Wnt to activate that Wnt and try to dorsalize the forebrain into a cerebral cortex. So this is more or less how it looks. So basically we start with our 2D epiblast stem cells. We put them in a V-shaped well. Within 24 hours, these cells will aggregate to each other. They will form these 3D embryonic bodies that you can see here. And then we just let them on matrigel within the specific media and put them to shake. And as like the culture happens and this happen, these organoids will keep on getting like bigger, bigger, and bigger, as you can see here in this day 8 organoids. And importantly, as you can see in these images, they do not just get bigger, but they do have a certain organization. You can see that here at the center of the organoid, we have a lumen and emptiness, on top of which a lot of cells are sitting on. So what are those cells? So this is an image of a day 4 neural organoid. And what you will be able to see is that we have a complete acquisition of a picobasal polarity, meaning that all of the apical membrane labeled by ZO1 is within the inner part of the organoid, nothing is outside. On top of this apical membrane, we have the pH3 positive neuronal progenitors, and these pH3 positive neuronal progenitors extend their fibers, labeled with nestin, towards the basal lamina. So clearly recreating the development of a normal neuroepithelia. But then the question is this really a cortex or is it any other place of the brain? So for that, we did some other more stainings. Here I'm showing you a cortical organoid at day 8. And as you can see here, we do recreate within this lumen that all of the progenitors are sitting on. They are PAX6 positive, such as it happens in the cortex, and they start to give rise to TBR1 positive deep layer cortical neurons that are like the fate that they will give rise to early during development. Then we can take this organoid further for more days. And by day 12, we will start to have this nice neuroepithelias arising in our system, where you can see that we have once again these PAX6 progenitors that on top have TBR2 red, labeled in red intermediate progenitors, and then they keep on giving rise to this TBR1 or CTIP2, that is another marker, postmitotic deep layer cortical neurons. So it's great. We have a great system that recapitulates the development of the cortex. The next question was like, is this protocol strong enough or efficient enough as to work with these 4 different F1 hybrid cells that we want to apply it to? And the short answer is yes. So here I'm just showing you some images of day 13 cortical organoids created from or arising from EPSCs from the 4 different hybrids, so the BLAC6 spread, BLAC6 small, BLAC6 cast. And as hopefully you can appreciate,
1 minute warning.
We have a huge amount of deep layer cortical neurons that are CTIP2 positive, showing that indeed our protocol is reproducible across batches, and it also works with highly divergent backgrounds. So the next thing that we did is single-cell RNA-seq to prove that that identity is true also at the transcriptomic level. And here I'm basically showing you each dot represents a single cell, and they're colored by the fate that they have. And indeed, that's the case. So as the single cell was proving, we have different clusters that give rise to radial glial cells, those cortical progenitors. We have another cluster, intermediate progenitor cells, that have the classical gene expression of TBR2 expressing neurons with neurogenin 2 and so on. And what is important about the single cell analysis is that they also showed that when we were to use the protocol with the 4 different F1 lines, they were giving rise to exactly the same fates. So it's reproducible, but then the last question that we had is like, so the brain in vivo keeps on developing. So it does not only give rise to deep layer neurons, but it will also generate upper layer neurons and astrocytes as time progresses. And these astrocytes are actually labeled by GFAP. And what I could see is that when we were to mature the organoid for a few more days, up until day 16, how could I start to see these GFAP positive astrocyte looking cells, suggesting that we do not only recapitulate early development, but we also recapitulate the succession of events that happens during brain development. And this is key because, like, if we are to compare this to, for example, like normal human brain organoid systems, they take as much as 100 days to give rise to these GFAP positive cells. We take 16 days to do so. So our systems are about like 6 times faster than the human ones. So in conclusion, hopefully I've convinced you that we've come out with a good in vitro system to recreate the way that the cortex is formed in vivo. And of course, my main goal right now is to use this with F1 hybrid stem cells and map those allelic imbalances across the wide variety of fates that we're obtaining. And so as through our development goes by. But I wouldn't like to miss this opportunity to also mention that we have not only done this cortical organoid protocol, but at the same time, we use these cells to generate a wide variety of organoids. So for example, hippocampal organoids, melanocyte-containing non-neural ectodermal organoids, some other organoids that give rise or recreate the development of the striatum. And importantly, these are highly reproducible. They work with all of our F1 stem cell lines, and they are so much faster than human protocols, so highly amenable to high throughput screenings, for example. And just as a whole, all of this is actually showing, kind of as a retrospective, that the epiblast stem cells, those stem cells that we're using to create all of that, are indeed a great tool to mimic development and to see how different structures arise. And I would like to finish neither without saying that, of course, we're using these technologies because they are useful for us, but it will be so much better if they could be useful also for the community as a whole. So please do not hesitate to contact us, email us, to asking for those cells, asking for those protocols or whatsoever, because we really want to share everything that we're doing. It will be so much better if people could apply them as well to their model system. And that's it. I just want to thank once again Thomas Verbuchen, who has directed all of these works, Zarina, Tarek, Marty, Jaylin, everybody from the lab, New York Stem for the founding, SDB for letting me present, and to everybody that was listening to this presentation. And I'll be happy to take any questions.
Thank you, Daniel, so much for that really interesting talk. I actually have a few questions while everyone is posting their other questions in the chat. So have you ever noticed any trends in chromatin accessibility, whether the allele is maternal or paternal?
Yeah, there's a lot of imprinting going on. Yeah. It totally happens. Like the cell lines have already some imprinting phenomena that we are aware, because like we look through many different cell types and the imprinting is always the same. So it's clear that there's something coming from the beginning already.
Oh, great. And then if you actually isolate epi-SCs from different genders of mice, do you actually see differences? So that's not just whether it's maternal or paternal, but if you actually have SCs taken from male or female mice, do you see any differences in the cis-regulatory variation in addition to genotype?
I would expect them to be. I do not know, but I would expect them to be some like imprinting related differences and like Y chromosome related type of things for sure.
Fantastic. So we also have a question in the chat from Augusto Granillo. It says, cool talk. I noticed on your volcano plot that there are more genes upregulated in the BLAC6 background rather than in spread. Why do you think that is?
Yeah,
and then do you find a similar thing with the other hybrids you've built?
So that's most notorious with the spread one. And that is actually, we are trying to look into what is that cost for. Like we are confident in our mapping strategies and we believe that they're working. But so that we want to look further into what could be the cause of that slight imbalance towards more one side than the other. What is important though is like at the bulk expression, like everything that we find, we validate in bulk RNA-seq and bulk attack and many different like experiments and so on. So like for the nanoc, for example, we're really sure that that is happening kind of thing.
Thank you. And Marcia Lucas would like to know, how long do the BLAC6 spread hybrids live?
I actually don't know. It's the question like whether it will be something in between, no? I mean, they have issues. Like, for example, the males are not even fertile. The females, yes. So I don't know if that comes with like some other issues that may impact their lifespan or something like that.
So you showed that you were using BLAC6 always as the female, and you mentioned that you have to. Is that just for controlling the experiment, or is that just because the offspring are inviable if you do the other reciprocal cross?
It's actually because it's easier to breed. Like at the breeding moment, if the female is the BLAC6, it will make things easier. Still complicated, but easier than if it was a male.
Well, I think those are all of the questions that we have. So we can then move on to the second half of the seminar. Thank you so much, Daniel, for presenting your work.
Thank you.
All right, thank you, Daniel, and thank you, Dana, for such wonderful talk. I want to ask you all to join me. It's my pleasure to introduce our next speaker, Dr. Jiae Lee. Jiae earned her bachelor's and her master's degree in biology from Yonsei University in Seoul, Korea in 2005 and 2007, respectively. Her master's work involved the use of using a proteomic approach to discover the role of glycosylation in modifying heat shock protein activity in both Drosophila and pancreatic beta cells. After completing her master's, Jiae moved to the University of Washington in Seattle, where she earned her doctorate degree in 2016 in the Department of Biology. There, she began work on dendrite growth and placement during Drosophila fruit fly development, which included discoveries on the protein RAW. After earning her doctorate, she stayed in Seattle and moved to the Department of Biochemistry to the Quan Lab, where she started to work on understanding the cellular mechanisms of dissemination during tumorigenesis, including the role of extracellular vesicles in this process. Please welcome Jiae as she shares her talk, her latest work and talk today on the systemic propagation of cytosolic DNA response via generation of large extracellular vesicles.
Thank you, Joseph, for the kind introduction, and good afternoon, everybody. I'd like to thank first SDB for inviting me, and I really appreciate this opportunity to share my work about extracellular vesicles that are produced from tumors that are made in developing tissue. So when it comes to understanding the complicated biological processes, it is somewhat like solving a word puzzle. You can get clues from pieces of information from previous studies in various contexts, but you'll need to connect them all using an appropriate model to understand the big picture. Today, I will be talking about a model I established for extracellular vesicles produced from invasive tumor cells. Extracellular vesicles, or EVs, were originally thought to be a source of cellular dumping or a way of cells to get rid of a needed material. But recently, EVs are emerging as an important mechanism for intracellular communication, so therefore being thought as a promising therapeutic target. For example, cancer cells disperse EVs to the surrounding environment, and EVs can transport information from cancer cells to other cells and vice versa, serving as an important mechanism of communication in the tumor microenvironment. So having an in vivo tumor EV model can be a unique opportunity to study this very complicated process. There are several different mechanisms of how EVs can be produced, such as from exosomes that are produced from multivesicular bodies, or microvesicles, or ectosomes, or large oncosomes, all produced from the direct budding of the plasma membrane. And EVs have spherical structure consisting of a lipid bilayer, and it can contain complex contents, including proteins, mRNA, microRNA, non-coding RNA, DNA, and specifically for oncosomes that are made from tumor cells, they can carry oncogenic cargoes. And intriguingly, the protrusions, blebs, and oncosomes are implicated in the intravasation and metastasis of human cancer. Therefore, understanding the mechanism of the formation and function of extracellular vesicles derived from tumor cell would be very crucial. To characterize the EVs derived from tumor cells, our group utilized an invasive tumor model developed in the developing fly larval system, induced in the developing eye antennal disc. Insect larvae have multiple sac-like epithelial structures called imaginal discs, which are the presumptive adult organs that will develop into eye, wings, legs, so forth during metamorphosis. In these kind of discs, when oncogenic mutations like RAS V12 mutation is accompanied with cell polarity gene mutation, like Scribble, then the cells transform into malignant tumor that overproliferates in the eye disc and metastasize into other body parts, such as the ventral nerve cord. And from its establishment in 2003, eye antennal disc tumor system has been widely used as an inducible metastatic tumor model. So first, I wanted to ask, will the eye antennal disc tumor produce the extracellular vesicles? I used RAS V12 itself as a control that induces hyperplasia, but it has no malignancy. And the tumor does not make the extracellular vesicles by ex vivo live imaging, but the RAS V12 Scribble mutation induces malignant tumor, and excitingly, the eye disc tumor produced tons of extracellular vesicles. I can image the production of the extracellular vesicles in higher resolution, and the number of vesicles can be counted and quantified, as well as the size, also quantified. And it had a size range of about 2 microns in average, which is a strikingly bigger size than the widely studied exosomes that is derived from the multivesicular body. I wanted to double check if the EVs are produced in vivo. So I did a whole mount larval live imaging, taking advantage that the larva is transparent. So left is a stitched image of a whole mounted larva that is induced with eye disc tumor on the top right. The tumor cells are the only cells expressing GFP, so you can hopefully see the GFP particles dispersed all throughout the body. And on the middle, it's a movie focused on the yellow box region, and here you can see that there are indeed robust EV formation also happening in vivo. And right, this is a control for the RAS V12 only disc that there is no EV formation. So in conclusion, RAS V12 Scribble eye disc tumor produces extracellular vesicles ex vivo and in vivo. To investigate what are the roles of the tumor EVs, the first thing to do is to isolate and collect the EVs. I established a simple protocol to efficiently isolate and concentrate the tumor EVs by differential centrifugation. And this accumulation of EVs is verified by fluorescent imaging, and the size were also quantified with a particle analyzer. To characterize which type of EVs these are, I examined the ultrastructure of the discs and the EVs. So compared to the control eye disc, the surface of the tumor eye disc is rough and irregular. The ECM layer that is shown pointed by the arrow in the control disc, the ECM layer is dissolved. And we could also see the EVs were produced directly from the plasma membrane, suggesting the EVs can be categorized as microvesicles or ectosomes. The isolated EVs look like a deflated soccer ball, which is a typical shape of an EV under TEM. So what would be the physiological role of these tumor extracellular vesicles? Tumors communicate with the rest of the body via various secreted factors that results in various detrimental defects in the body. And one of the major response is increased inflammation. So we wanted to ask, will tumor extracellular vesicle induce systemic immune response in the larva? So in flies, fat body is the central organ responsible for humoral immune response through conserved pathways for synthesizing and secreting antimicrobial peptides into the hemolymph. These peptides are like the inflammatory cytokines in mammals. So I established a system that I first collect the EVs from disc tumors and inject them using a microinjector device into a naive host, anticipating the immune response would be elevated in the fat body. I can measure the systemic immune response by observing GFP reporter activity or perform qPCR on the immune response genes for more precise measurement. So I injected the EVs, did the experiment, and then did the assay in the fat body. And for the first method for GFP live reporter activation, I compared with various controls, compared with no control, no injection, PBS injection, EVs collected from the control disc, the RAS-only disc, and finally the RAS-scrubal tumor disc, which was the only group that showed significant induction of the reporter activity. I also measured the mRNA expression of the immune response genes, 3 different kinds here. And indeed, the RAS V12 disc-derived EV fraction injection was the only, was sufficient to increase the immune response in the fat body, innate immune response in the naive host, and that does not have a tumor. Okay, so the EVs are being released into the hemolymph, the circulating body fluid, and in hemolymph, there are circulating immune cells, the hemocytes. So next, I asked if the hemocytes interact with these EVs. From the isolated hemocytes that are labeled in red, indeed, I could image GFP vesicles that are been engulfed by the hemocytes. And I also, then next I asked, are these hemocytes necessary for the immune response that happened in the fat body? So for that, I performed genetic ablation of the hemocytes in the larva. The hemocytes are labeled with GFP, and it didn't induce apoptosis by expressing a gene called Reaper, and confirmed that the genetic ablation completely ablated the GFP signal, meaning that the hemocytes are all gone. And indeed, by injecting the tumor-derived EVs in the control, increased the immune response gene expression, but the increase of those genes have been reduced by the ablation of these hemocytes. So, which means that ablation of the hemocytes, so the hemocytes are necessary for the host immune response in the fat body. So then what could be the mechanism of EV formation in the metastatic tumor? I hypothesized the cytosolic DNA sensing cGAS STING signaling pathway is responsible for the EV formation. So cGAS is an innate immune sensor that recognizes cytosolic double-stranded DNA. And unlike normal cells, cancer cells are often filled with cytosolic double-stranded DNA. Recent studies suggest that chromosomal instability, or CIN, is a primary source of cytosolic double-stranded DNA in tumor cells, presumably because the tumor cells go through rapid division and they can override checkpoints. The cytosolic exposure of genomic contents triggers the cGAS, cGAMP, STING pathway, and STING can mediate the transcriptional activity of a broad repertoire of molecular programs, which include inflammation, autophagy, metastasis, etc. So first, I asked if I can observe the chromosomal instability in the disc tumor cells. The nuclear envelope is labeled with lamin in magenta color, and yes, I could see hallmarks of CIN, such as nuclear envelope deformation, chromosomal leak, chromatin bridge formation, and micronuclei formation. Moreover, the transcriptional level of the STING pathway gene, the downstream genes in the tumor disc, was elevated in the RAS-Gribble tumor discs. Now, when the central gene, the STING, was knocked down by 3 different RNAi lines, the expression of these downstream genes were dropped accordingly, which means that the STING pathway genes are elevated in the disc tumor. Now, next question would be, will this STING knockdown in the RAS-Gribble disc, then will it inhibit the EV production? So the RAS-Gribble disc produces very robust EVs, but in the STING RNAi line, here I'm showing 2 different lines, they have very few EV formation, and the number of EV formation can be quantified. So STING in the tumor cells is necessary for EV production. Now let's draw this full circle. If we induce RNAi for STING in the tumor, will it be sufficient to reduce the immune response in the fat body? And yes, immune response in the fat body is suppressed by the STING knockdown in the tumor. So STING is necessary in the tumors for activating systemic immune response. Remember, there is one more player in this field, the hemocytes, that the hemocytes were necessary for carrying out the systemic immune response. And we already saw the hemocytes are engulfing the tumor EVs, which can be relaying the STING signaling and elevate the STING signaling in the hemocytes. So we hypothesize that STING in the hemocytes might be required for transducing the signal mediated by large EVs to the fat body. To test this model, we first examined whether injection of EV fractions from RASV12 STING disc could increase the STING signaling in the hemocytes. We also confirmed this by inducing STING RNAi in the hemocytes. Now, EV injection did elevate STING signaling in the hemocytes, which also decreased back by STING RNAi expression in the hemocytes. This pattern was consistent throughout the other STING pathway genes. So STING signaling is indeed elevated in the hemocytes by tumor EV, encountering the tumor EVs. So next, we confirmed whether STING in the hemocytes was really important for the EV-induced systemic immune response in the fat body. So now we are manipulating STING in the hemocytes and then measuring the immune response in the fat body. And after injection of the tumor EV, the host that STING is knocked down in the hemocytes indeed have a lower elevation of the immune response genes in all 3 different broad range of immune response genes. Now, these results altogether indicate that STING in the hemocyte is required for the systemic immune response induced by the tumor EVs, and indicates also that the tumor-derived STING pathway is systemically propagating by the large extracellular vesicles. Before wrapping up, I'd like to share the exciting results from a postdoc in our lab, complementing the findings from my Drosophila model. Blebbing and EV formation is also seen in human cancer cell culture system. So the size of the tumor EVs are comparable to the fly eye disc tumor EVs. Now, excitingly, the activation of STING pathway could increase tumor EV production by transfecting double-stranded DNA and inhibiting transfecting the inhibitors of STING can suppress the EV formation in these cells. And more intriguingly, we could see the tumor EVs are containing the STING pathway components, such as CS shown here, and also it can contain DNA. And this is especially interesting because it reconfirms that tumor EVs can be used for propagating the STING pathway. And I think this is a very good example showing that the discoveries made in Drosophila in vivo model can be expanded to mammalian tissue culture systems in other contexts. And these approaches from different systems can synergistically advance our understanding on the functions of tumor-derived EVs. So as a short summary overall, so I have shown you that the malignant tumor in the eye disc produces large extracellular vesicles. The tumor-derived EVs can induce systemic immune response in the fat body. And hemocytes are necessary for these systemic immune response. There are chromosomal instability are occurring in tumors, which activates the STING signaling pathway. The STING signaling is necessary for EV production and systemic immune response in fat body. And I have also shown you, finally, the STING signaling also in the hemocytes is necessary for the systemic immune response in the fat body. And overall, the tumor EV injection model opens many opportunities to study the mechanism of EV production and function, not only in the tumor context, but also in normal physiological conditions, because I can inject the tumor-derived EVs into a naive host. So I'd like to thank our lab, especially my PI, Young, and Hyungjun Park, the postdoc that is working on the mammalian cell culture side of the project, the rest of our lab, and the fly community for providing very generous resources and my funding sources. And with that, I'll be happy to take any questions.
Thank you, Jiae. That was a wonderful talk. We have one question already, so I encourage all of you to type in your questions, but let's start with the first question from an anonymous attendee. It says, hi, I'm an undergrad. This talk was very intriguing. Could CRISPR-Cas9 be used in order to extract the STING pathway, which produces EVs from metastatic tissue? I'm also wondering whether the EVs from metastatic tissue could be used in stem cells because the eye antennal disc tumor can produce EV in vivo as well as ex vivo.
Yeah, thank you. That's an awesome question. So the first part of the question is, so the first part of the question is asking why did I can use CRISPR-Cas9 to completely knock out STING instead of using an RNAi line. Of course, I can do that. I just didn't do that because there is a complicated genetic tool making for doing that, and inducing RNAi is more easier. But indeed, I can do that to completely knock out the STING pathway genes, and that would be really, really cool to do. Actually, actually, I think that's a really great idea to dissect out which STING pathway downstream genes are actually being important in the following processes. So thank you for that idea. the next part of the question was whether the EVs from metastatic tissue in the stem cells could be used in stem cells. Sorry. Yeah. Okay. So in the stem cells, sure. So I think, of course, that is a great idea. This is not related to this project, but we have been observing that the intestinal stem cell, the system also can produce EVs. So we have been observing EV production in many different various contexts. So yeah, thank you for that idea. Very good insight. But haven't been really dug in the functions or the outcomes of those EVs and what is different from the tumor-derived EVs and so forth. So yeah, but thank you for that question.
What a very great question from an undergrad. I really love
Absolutely,
it. Somebody's
love it.
really interested. All right, so let's move on to the second question from Katie Ong, who first says, excellent talk, for which I agree. The question is, for the EVs that you purify for injections, are they isolated from discs that are cultured ex vivo or directly from larval hemolymph? And after that, also, do you have a guess as to what signals from STING-activated hemocytes stimulate the immune response in the fat body?
That's an awesome question. So I apologize for going really quick on that protocol part, but I do ex vivo incubation in the live imaging media, essentially to keep them alive. And then I do purification processes, but they are dissected out and then and incubated for 4 hours in room temperature. And I do the purification. So yeah, that's how I collect my EVs. I would acknowledge that my EVs that I collect is a very heterogeneous population. So, but at least it is sufficient to carry out the subsequent immune responses. The next part of the question was what signals from the STING-activated hemocytes are stimulating the immune response in the fat body? That is a really great question that I want to address, of course, that what is this part? And here I have the ... means that I don't know very well at this point, but from this injection system, I think it will be, this is a great opportunity to find out which signaling is actually elevated in the hemocyte. So I have a plan to assess the transcriptome profiling in the hemocytes that have engulfed the EVs versus not engulfed EVs and so forth. So yeah, that would be the next step indeed.
Right, that's great. So we're going to move on to the next question from Michelle Stars-Guyano, who says, hi, great talk. I was wondering if you tried to induce EV formation by ectopically activating the STING pathway, maybe in a RAS V12, but not in Scribble mutant. In other words, is the STING pathway sufficient for EV release?
That is also a very good question. I haven't done that experiment, but I indeed can do it. So that would also be my next step. It is the way that I am inducing these tumors, and it's not the method that I developed. It had been used for 20 years of this metastatic tumor model in the eye disc, is that you induce a clone. You induce a clone to get these aggressive tumor growth. And to do that, you have to do a lot of recombinations on the chromosome and so forth. But I can make it, and the next step would definitely be testing for the sufficiency of STING pathway in the non-EV forming tumor. So here I only tested the necessity of the STING signaling in the tumors, but I haven't done the other way. But that's a great idea. Definitely need to do that. It's on my laundry list. But thank you for the question.
Yes, that was a great question. And since we have a little bit of time before we wrap up, since Scribble is my favorite protein,
awesome.
I was also wondering if you have tried to induce any of the clones with its binding partners, DisLarge or LGL, and if you see similar results in those whether it's specific to scribble or polarity loss in general?
So that's a great question. The short answer is I haven't looked at it before, but we know that, the DLG or LGL, those are sufficient to induce the malignant type of tumor. So that is one direction I want to go through. I also want to induce P35 to block the apoptosis or the elimination of scribble only tumor to see if that will be sufficient to induce the EVs and not even having the RAS V12. So that is also the other direction that I'm really interested in.
All right, thank you. And if there are no more questions, I think we can go ahead and start to wrap this up. So please join me in thanking Daniel Medina Cano and Jiae Lee for their excellent talks. Thank you, Dana, for helping me host this. And thank you to SDB for allowing the opportunity for all of the speakers this year. The seminar has been recorded and will be available on the SDB website next week. Please don't forget to join us. Mark your calendars for next month's seminar on Friday, March 10th, 2023, where we will hear from Lauren Walker from the University of Pennsylvania and Zhengfen Xu from the University of Michigan, who will present their current work. Thank you all for joining us today.